Hello everybody, welcome to the T.Y.B.Sc class. Today is the third lecture of the course 3 MB333 Enzymology. We have started studying the topic 1, namely enzymes. To be very precise, we are studying the unit 1 of the topic 1, structure of enzymes. And in this topic, we are going to study the methods that are used to determine the amino acids at the active site of an enzyme. The amino acids at the active site of an enzyme can be studied by two methods, physical methods and chemical methods. All the chemical methods to map the amino acids at the active site we have already studied in the earlier two lectures. Today we shall study X-ray crystallography as a physical method to study or to determine the amino acid residues at the active site. The second physical method, nuclear magnetic resonance, we shall study in the next lecture. The learning objectives of this particular lecture can be defined as the student at the end of the lecture or by the end of the lecture should be able to describe the active site mapping with X-ray crystallography with respect to the principle, procedure, and applications of this particular method. To introduce X-ray crystallography, which is a tool to study the three-dimensional molecular structure of a protein crystal. X-rays are particularly selected to study the crystals from amongst all the rest of the rays just because they have a wavelength similar to the size of the atoms that are present in a protein crystal. A beam of X-ray strikes the protein crystal and it is diffracted into many different directions or many specific directions. Then, from the angles at which the X-rays are diffracted and from the intensities of the rays that are striking the X-ray film, it is the crystallographer who analyzes or studies the angles and the intensities, and from this he constructs a three-dimensional image of the density of electrons that are present in the crystal. To go back to the history of the development of X-ray crystallography and to be grateful to the scientists who have contributed to this wonderful technique, which has helped us to study the enzymes and the catalytic behavior or the catalytic mechanism of the enzyme. To start with, it was Röntgen who discovered X-rays and was awarded a Nobel Prize for this contribution in physics in 1901. Then in the row is Max van Leeuwen, who studied that X-rays are diffracted by proteins. So he studied the diffraction of X-rays by the protein crystals, and for this contribution he also was awarded a Nobel Prize in Physics in 1914. Then comes the father and son duo, together called as William Bragg's, with their Bragg's equation, which they constructed for the constructed by analyzing the diffraction pattern of X-rays by the crystals. And for this contribution towards X-ray crystallography, they also were awarded with a Nobel Prize in Physics in 1950. The application of X-ray diffraction studies in various scientific fields have led to a total of about 29 Nobel Prizes so far and many more to come. The successful use of crystallography in many scientific fields was recognized by the United Nations, which declared 2014 as the International Year of Crystallography. The principle of the X-ray crystallography is nothing but the diffraction of a beam of X-rays by the electrons that are present in the atoms of the proteins and then studying this diffraction pattern and with the help of a computer software constructing a three-dimensional image of the protein crystals. The components that are needed for X-ray crystallography are a source which generates the X-rays, a protein crystal which serves as a sample, and a detector. The step one of the procedure is you first need to crystallize a protein. For crystallizing a protein, it is very much necessary to have a purified sample. The crystal has to be sufficiently large, and this is the basic requirement and a basic limitation also of crystallography, X-ray crystallography. The size of the crystal should be more than zero point 0.1 millimeters. And to have a crystal having a size more than zero point 0.1 millimeters, you need quite a large amount of the purified protein, which should be at least 10 milligrams per ml. The protein crystal is then mounted on in a capillary and is exposed to a beam of X-rays having a wavelength of 1.54 angstrom units. The wavelength, the range of wavelength which constitutes the X-rays is around 10 raised to minus 8 meters to 10 raised to minus 11 meters, and these are shorter than the rays of the visible light. From this, for the X-ray crystallography, we need a monochromatic beam of X-rays. That is, we need to use a filter which will allow to pass through it the beam of X-rays having a single wavelength or having a same wavelength. So such monochromatic beam of light is then passed through the protein crystal. The crystal is rotated so that all the sides of the crystal can be studied, and then the intensity of every spot is recorded at every orientation of the crystal. In this way, the multiple data sets of these studies are collected. The X-ray beam hits the crystal. Some rays pass through the crystal, while some rays are scattered in various directions from the external environment or from outside of the crystal. The X-ray is diffracted by the atoms that are present in the protein crystal. The X-ray beam is diffracted into many specific directions, which depends upon the arrangement and number of electrons that are present in the atoms of that particular crystal. The X-ray beam then hits the diffracted X-ray beam, you can say, then hits the X-ray film and forms black spots that are proportional to the intensity of the scattered X-ray beam. This forms a pattern which is called as the X-ray diffraction pattern. To illustrate the same procedure now, here is the source which generates the X-ray beam. The X-ray beam then passes through a filter which generates a monochromatic light. The monochromatic light is then focused on this crystal which is mounted on the capillary. The atoms that are present in this crystal, depending upon the number of electrons that are present in the external orbitals of these atoms, then diffract the X-ray beam in different directions. And here you can see the different intensities and the different sizes of the black spots that are formed on the X-ray film. Electrons that are present around the nucleus of each atom, they scatter the X-rays to different amplitudes. So here just to make you revise about what is the amplitude, I have put this wave structure. So this is the amplitude and this is the wavelength as we have seen. So each atom contributes to the scattering of the beam or contributes to the scattered beam. The amplitude of a wave scattered by different atoms changes according to the number of electrons that are present in that atom. So a carbon atom, it scatters the wave 6 times stronger than that which is scattered by the hydrogen atom. And this leads to the differences in the intensities and the sizes of the spots that are formed by the diffracted X-rays which hit the X-ray film. The third step is now analysis of the diffraction pattern, wherein the intensity of each spot on the X-ray film is measured and interpreted by a mathematical treatment called Fourier transform system. The computer process or a computer software then processes this data and constructs the structure of the atom or the molecule in the crystal. So a three-dimensional crystal structure is thus created. This model of the atomic arrangement is called the crystal structure and is stored in a public database. Uses of crystal, as we have seen earlier, X-ray crystallography has been used in many different fields, and its use in many different fields has fetched more than 29 Nobel Prizes. So let us see where all it can be used. It is used to study the materials which can form crystals, which is the basic requirement of X-ray crystallography. So all the substances like salts, metals, minerals, semiconductors, various inorganic, organic, and biological molecules can be studied using X-ray crystallography. X-ray crystallography gives us the mean positions of the atoms in the crystal. It is used to find the size of the atoms and types of chemical bonds in the substance or the crystal that is scanned with X-rays. In this way, it has helped us to study or it has helped us to reveal the structure of many biological molecules like vitamins, drugs, proteins, and DNA. X-ray crystallography was first used to determine the protein structure in late 50s. So just to see what we have studied earlier in the earlier slides, we have seen that the Bragg duo, the father and son duo, they have bagged the Nobel Prize in 1915 for analyzing the X-ray diffraction pattern of crystals. But just imagine that it took 35 more years for X-ray crystallography after its discovery to be used in the field of biological sciences. Anyway, we are grateful to them. The first three-dimensional crystal structure of an enzyme which was determined by X-ray crystallography was the hen egg white enzyme, which is called as the lysozyme. This technique has a lot of limitations also, which can be summarized as follows. It needs an extremely pure protein sample. Purification of protein is also a very difficult process and it needs a large number of efforts to purify a particular protein. Secondly, the protein to be crystallized, it needs a large sample because you need a larger sized crystal. And so you need to purify a very large amount of protein, which again adds to constraints in terms of the amount and the amount of money that you need to purify the proteins and the labor and the glassware that is involved in it. But many proteins cannot be studied by X-ray crystallography because they cannot be crystallized. That is, they aren't amenable to crystallization at all. They cannot be crystallized. And so such proteins need to be studied by the other methods of mapping the amino acids at the active site of the enzyme. So thank you, students. I'm very sure you have understood X-ray crystallography as a technique and the application of X-ray crystallography in the field of biological sciences, especially to study the enzyme structure of the enzymes that we need to study for their catalytic applications. Thank you so much.